Hello everyone. Our lesson for today is about protein sorting and transport into organelles. In this lesson, you will be able to learn about the molecular mechanisms of how proteins are sorted and transported to their respective destination organelles. Specifically, this lesson will discuss the different types of protein transport mechanisms, then focus more on gated transport to the nucleus and transmembrane transport to other cellular organelles. At the end of this lesson, you are expected to differentiate the types of protein transport mechanisms, identify sorting signals and their complementary sorting receptors, discuss the process of protein transport to the different cellular compartments, and finally identify the components of gated and transmembrane transport systems. The motivation question for this lesson is this. Is it possible for cellular compartments to acquire proteins that are synthesized in the cytosol? So I hope you will be able to answer this question at the end of this lesson. So all proteins begins their synthesis on ribosomes in the cytosol. So in the previous lesson, we've talked about transcription and translation. So we know that the translation is where the mRNA is expressed or translated into protein sequences or polypeptide. So we know that ribosomes could occur as a free living in the cytoplasm, or they are free within the cytoplasm, but some of these are attached to the endoplasmic reticulum. That's why some of the ERs are called rough endoplasmic reticulum because of the attached ribosome. And synthesis of protein could also occur there. Okay. Although there are also some proteins that are synthesized in the mitochondria and plastids because they have their own ribosomes as well. So the subsequent fate or future or the depends on their amino acid sequence. So it is already embedded. It's already in the sequence of the protein synthesized. No, that determines where will it be transported. So these proteins contain sorting signals that will direct their delivery to locations outside the cytosol or to organelle surfaces. Some proteins do not have sorting signals. That's why they just remain in the cytosol as permanent residents. Because you cannot transport a protein if they have no sorting signal, because the process wouldn't know where to send the particular protein if they have no particular specific sorting signal for a particular compartment or organelle. Many others, however, have specific sorting signals that direct their transport from the cytosol to the nucleus, the ER, the mitochondria, plastids, or even at the peroxisomes. Sorting signals can also direct the transfer of proteins from the ER endoplasmic reticulum to other destinations in the cell. So to understand the general principles by which sorting signals operate, it is important to distinguish three fundamentally different ways by which proteins move from one compartment to another. So you have here an example of an unfolded protein. So this is a newly synthesized protein, and if you notice here, at the end terminal of the of this protein, you can find some signal sequence or sorting signals. So they're just the same, the sorting signals and the signal sequence. Now some could be located, some of these signal sequences or sorting signals could be located at the ends of the linear protein, while others could occur or distributed throughout the protein itself. So they produce a signal patches. So this is an example of a signal patch, and this is the signal sequence at the end of the protein. Okay, so the first, you have here the fate of the protein. So we know the protein synthesized in the ribosomes in the cytoplasm could be transported to the nucleus, mitochondria, chloroplasts, or peroxisomes. Okay, while those proteins that are synthesized in the ER, okay, could be transported through nuclear membrane, plasma membrane, secretory vesicles, in the endosomes, into the lysosomes, and at the peroxisomal membrane. So, as we've said earlier, there are three ways by which proteins are transported from one location or compartment of the cell to another. So, what are these three mechanisms? It could be gated transport, transmembrane transport, and vesicular transport. Okay, let's talk first about gated transport. So proteins and RNA molecules move between the cytosol and the nucleus through the nuclear pore complexes in the nuclear envelope. So the nuclear pore complexes, okay, so this is an example of a gated transport. You have here the nuclear pore complex, okay. So yeah, the cytosol and the nucleus is the nuclear pore complex. They function as selective gates that support the active transport of specific macromolecules and molecular assemblies between the two topologically equivalent spaces, although they also allow free diffusion of some molecules, but no, that depends on the size of the molecule. So the smaller, the easier for them to diffuse through the nuclear pore complex. The bigger, so it will be difficult for them. Another type of transport mechanism is a transmembrane transport. Transmembrane protein translocators, okay, they are directly transport specific proteins across the membrane from the cytosol, okay, into the topologically distinct space. Now, do not confuse yourselves again with the term cytosol and cytoplasm. We know that the cytoplasm are all the materials that you can find inside the cell. Cytosol is the liquid part, of the fluid part of that cytoplasm. So the initial transport of selected proteins from the cytosol into the ER lumen or mitochondria, for example, occurs through transmembrane transport. So transmembrane meaning there is transfer or translocation through a membrane. Okay, transmembrane. If the transport is to the mitochondria, we know that there is a nuclear complex that acts as a gate, then we call it the gated transport. Okay, so if it is passing through a membrane, that is transmembrane transport. Integral membrane proteins, on the other hand, often use the same translocators, but translocate only partially across the membrane. Okay, just like this. So the protein becomes embedded in the lipid bilayer in the process. The third type is the vesicular transport. Membrane enclosed transport intermediates, which may be small, okay, just like this. The spherical transport vesicles are larger, irregularly shaped organelle fragments. They ferry the protein, or they transfer the proteins from one topological equivalent compartment to another. So the transport vesicles and fragments become loaded with a cargo of molecules that is derived from the lumen of one compartment as they bud and they pinch off from the membrane. So they discharge their cargo in the second compartment by fusing with the membrane enclosing the compartment. The transfer of soluble proteins from the ER to the Golgi apparatus, for example, occurs in this way. So because transported proteins do not cross a membrane, vesicular transport can move proteins in between topological equivalent compartments only, like the ER, the Golgi, and outside of the cell. So they do not cross the membrane. They are transported by a vesicle that will pinch, that will bud off, and will fuse with the other compartments. We will talk more about vesicular transport in our next lesson. So for this lesson, we will be focusing more on gated transport and transmembrane transport. So in all of these processes, the signal sequences or the sorting signals and the sorting receptors are very important. So each mode of protein transfer is usually guided by sorting signals in the transported protein, which are recognized by the complementary sorting receptors. So you have here, for example, protein import into the endoplasmic reticulum. So you have here the protein synthesized have a specific signal sequence of the sorting signal to the ER only. So once it is recognized by the sorting receptor, the signal recognition particle receptor, it will be transported through the membrane. Okay. So, if a large protein to be imported into the nucleus, for example, it must possess a sorting signal that receptor proteins recognize to guide it through the nuclear pore complex. And if a protein is to be transferred directly across the membrane, it must pass a sorting signal that the translocator, like this, could recognize. Otherwise, likewise, if a protein is to be loaded into a certain type of vesicle or retained in certain organelles, like in the vesicular transport, a complementary receptor in the appropriate membrane must recognize its sorting signal. So most protein signals involved in transmembrane transport reside in a stretch of amino acid sequence, typically 15 to 60 amino acid residues long. Such signal sequences are often found at the end terminus, at the amino end of the polypeptide chain. In many cases, specialized signal peptidases, the enzyme that removes the signal sequence from the finished protein once the sorting process is complete. So signal sequences can also be internal stretches of amino acid which remain part of the protein and are not cleaved. So such signals are used in gated transport into the nucleus. So most commonly found for gated transport into the nucleus are signal patches in the protein. So signal sorting signals can also be composed of multiple internal amino acid sequences that form a specific three-dimensional arrangement of the atoms on the protein surface. So these are called signal patches. They're used for nuclear import and sometimes in vesicular transport. So each of these signal sequences, they specify a particular destination in the cell. Okay, so different organelle in the cell have different sorting signal for a particular type of protein that uh, is attached to them. Okay, so for example, of a signal sequence that is important for import into the ER. So you have here, and for the lumen of the ER, for import into the mitochondria, import into nucleus, and import to the peroxisomes. Okay. So we will now talk about the gated transport, or the transport between the nucleus and the cytosol. So the nuclear envelope consists now of an inner, an inner and outer membrane. So you have here a double membrane. Okay? So inner membrane and the outer nuclear membrane, ONM, inner nuclear membrane. And this is actually continuous with the endoplasmic reticulum. So if you notice here, this is the lumen, which is continuous with the lumen of the ER. So actually, the outer membrane of the nucleus, of the outer nuclear membrane, is actually continuous with the membrane of the endoplasmic reticulum. Okay. So all the proteins that function in the nucleus are synthesized in the cytosol and are then imported. Okay? So the extensive traffic of materials between the nucleus and the cytosol occurs through the nuclear pore complexes, or the NPCs. So you have here. So they serve as the gateway to the nucleus. So these provide a direct passageway across the nuclear envelope. Now, each of these NPC, or nuclear pore complex, is composed of a set of proteins called the nucleoporins. Okay? Small molecules diffuse passively through the NPCs, but large molecules have to be actively transported. So proteins containing sorting signals called the nuclear localization signals are recognized by the nuclear import receptors. Okay, so you have here a protein with an import gene that is recognized by the receptors, and so it was able to be transported inside the nucleus. So some proteins, including the nuclear import and export receptors, they continually shuttle between the cytosol and the nucleus. So say for example here, particular protein that is taken inside can also be taken outside. So the monomeric enzyme called the GTPase run provides both the free energy, no? So this is the run GTPase. It provides both the free energy and the directionality for nuclear transport. So cells regulate the transport of nuclear proteins through the NPCs by controlling the access of these molecules to the transport machinery. So that was the goal for the gated transport, no? The transport from the cytosol to the nucleus. Now let us start our discussion with transmembrane transport. So first is the transport of proteins into mitochondria and chloroplasts. Although mitochondria and chloroplasts they have their genetic systems, so we know that they have their own genetic materials. To the mitochondria and the chloroplast genetic materials, they also have their own proteins because they can also produce their own proteins because they also have their own ribosomes. Okay. So, however, they only produce a small proportion of the proteins that they need, for their physiological process because through time during the evolution, some of these genes have been lost or some have been localized or transferred into the nuclear genome. So, in both cases, proteins are transported in an unfolded state across both outer and inner membranes simultaneously into the matrix space or the stroma. No? So both ATP hydrolysis and a membrane potential across the inner membrane drive translocation of these proteins to mitochondria, whereas GTP and hydrolysis drive translocation into the chloroplast. So there are what we call chaperone proteins in the cytosol of the cytosolic HSP70 family that maintain the precursor proteins in an unfolded state. It needs to be unfolded because it will be difficult for it to be transported through the membrane, across the membrane, if they are already folded in the state. So HSP binding will bind to the linear the structure of the newly synthesized protein so that it will not fold itself, because we know that one of uh, the secondary folding or the folding of the protein into secondary structures is dictated by the alpha the functional groups, as well as uh, the different groups that are attached to the alpha carbon. Okay, so in order for it to be protected so that the proteins will not fall, so HH370 will be bounded to it. So only proteins that contain a specific signal sequence are translocated across these membrane of mitochondrion and the chloroplast. The signal sequence can either be located at the end terminals and can be cleaved up after import, or be internal and could be retained. Transport into the inner membrane sometimes use a second hydrophobic signal sequence that is unmasked when the first signal sequence is removed. And this is especially true for transport into the chloroplast because they have the third membrane, which is the thylakoid membrane. Okay. So in chloroplast, import from the stroma into the thylakoid can occur by several routes, which we will discuss later. Okay. So this is how the proteins are transported into the mitochondria. So they have multi-subunit protein complexes that function as protein translocators, and the one that mediate movement across mitochondrial membranes. So these are the TOM, TIM, SAM, and OXA complexes. So the TOM complex transfers proteins across the membrane. The two TIM complexes, the TIM23 and the TIM22, okay, transfer proteins across the inner membrane. So if you notice here, the TOM is found outer membrane, TIM23 and TIM22 at the inner membrane of the mitochondrial membrane. So again, this is a double membrane component. So the TOM complex is required for the import of all nucleus-encoded mitochondrial proteins. It initially transfers the signal sequence, okay, into the intermembrane space of the the mitochondria. Okay. And during the protein import, the N-terminal signal sequence of the mitochondrial precursor protein is recognized by the receptors of the TOM complex. The protein is then translocated through the TIM23 complex so that it is transiently spans, so it temporarily spans both mitochondrial membrane. Okay? So the signal sequence is cleaved off, okay, by a signal peptidase in the matrix space, okay, to form a mature protein. So you have here the cleaving of the signal sequence. The free signal sequence is then rapidly degraded. So in the case of beta barrel proteins, this is a beta barrel protein, which are particularly abundant in the outer membranes. After transport through the TOM, what will happen is that it is passed on to an additional translocator, the SAM complex. So this helps them to fold properly in the outer membrane, forming the beta barrel protein. The TIM23 complexes transport some soluble proteins into the matrix space and helps to insert the transmembrane proteins into the inner membrane. The TIM22 complex mediates in the insertion of a subclass of inner membrane proteins, including the transporter, and moves the ADP, ATP, and phosphate in and out of mitochondria. Okay? So yet another important protein is important. Now this is the OXA, okay, the OXA protein. So the OXA, okay, mediates the insertion of those inner membrane proteins that are synthesized within the mitochondria. It also helps to insert some important inner membrane proteins that are initially transported into the matrix space by another complexes. Now, let us talk about the transport into the chloroplast. So protein transport into chloroplasts resemble transport into mitochondria as they are almost the same. However, the protein components that form the translocation complexes, they differ. Also, chloroplasts have an extra membrane enclosed compartment which is called the thylakoid. So aside from the outer, the inner membrane, on the inner membrane it also has its own membrane in the thylakoid. Many chloroplast proteins, including protein subunits in the photosynthetic system of the ATP synthase, are located in the thylakoid membrane. So the precursor proteins for these types of proteins contains an N-terminal chloroplast signal sequence, followed immediately by the thylakoid signal sequence. So the chloroplast signal sequence initiates translocation to the stroma by a mechanism similar to that used for the translocation of the mitochondrial precursor proteins into the matrix space, although the transloculator complexes, the top and the thick are different. So the signal sequence is first cleaved up, unmasking the thylakoid signal. So after the the protein containing the signal sequence is transported across the membrane, the signal sequence that directs it to the to the chloroplast will be cleaved up. In this way, the signal sequence that is masked here will be unmasked. So there's another signal sequence here that directs the transport into the thylakoid membrane. So translocation to the thylakoid space or thylakoid membrane can occur by any of the following routes: the SEC pathway, SRP-like pathway, the TATH pathway, and the spontaneous insertion pathway. So what are these, and how do they differ from each other? So the SEC pathway uses components that are homologs of SEC proteins, which mediate protein translocation across the bacterial plasma membrane. The SRP-like pathway, the SRP-like pathway uses a chloroplast homolog signal recognition particles, the SRP. The TAT pathway stands for twin arginine translocation. Now, the two arginines are critical in the signal sequences that direct proteins into this pathway, which depends on the proton gradient across the thylakoid membrane. The fourth one is a spontaneous insertion pathway, so it does not require any protein at all, it's just inserted across the thylakoid membrane. Now let us talk about another type of transmembrane transport, which is a transport of proteins into peroxisomes. So peroxisomes are specialized for carrying out the oxidation reactions using molecular oxygen. So they're the ones that generate hydrogen peroxide, which they employ for oxidative purposes that contain catalase to destroy the excess hydrogen peroxide. Okay, so like mitochondria and plastids, the chloroplasts, peroxisomes, however, all of their proteins are encoded in the cell nucleus, unlike the chloroplast and the mitochondria because they can also synthesize their own proteins because they have their own genetic material and ribosomes. But peroxisomes do not have that capacity, just like of the mitochondria and chloroplast. Some of the proteins are conveyed to peroxisomes by peroxisomal precursor vesicles that bud from the ER, but most are synthesized in the cytosol and directly imported. So transport of proteins into peroxisomes could be vesicular, not through vesicular transport, or it could be through transmembrane transport. Vesicular if they are from the ER and transported to the peroxisomes. Now, transmembrane if they are synthesized in the cytosol. So a specific sequence of 3 amino acids near the C-terminus of the many latter proteins functions as a peroxisomal import signal. So the mechanism of protein import differs from that of mitochondria and chloroplasts, in that even oligomeric proteins are imported from the cytosol without unfolding. So it is different because in other organelle transport, the HSP70 is attached so that it will not fold. But in here, they could be transported. The protein, even its folded form, could be transported without unfolding. Now let us talk about transport of proteins into endoplasmic reticulum, or the ER. So the endoplasmic reticulum, the extensive ER network, serves as a factory for the production of almost all of the cell's lipids. Okay. Also, a major portion of the cell's protein synthesis occurs in the cytosolic surface of the rough ER. Virtually all proteins destined for the secretion or for ER itself, the Golgi apparatus, the lysosomes, the endosomes, and the plasma membrane are first imported into the ER from the cytosol. Okay, so in the ER lumen, okay, in between the outer and the inner membrane, you call that the lumen. The proteins will fold and oligomerize. So the sulfide bonds now are formed and end-leg oligosaccharides are added. Actually, this is a very complex process. No, I have when I was doing my masters, we have taken this subject, no, during my physical chemistry and structural analysis of molecules na course. And it's for the whole semester, we've just talked about protein folding, no, in the ER. So for advanced molecular biology, I think it will be one of the topics na that will be discussed. But for now, no, what you need to remember is that once the proteins are transported to the ER lumen, it is where the modification of the product, now many things will happen, inside the lumen. So the pattern of end-link glycosylation is used to indicate the extent of protein folding, so that proteins leave the ER only when they are properly folded. So what happens if they are not properly folded? So those that do not fold or oligomerize correctly are translocated back into the cytosol where they are deglycosylated, polyubiquitinated, so that they could be degraded in the proteasomes. If misfolded proteins accumulate in excess in the ER, they trigger an unfolded protein response, which activates appropriate genes in the nucleus to help the ER cope, with this accumulation of unfolded proteins. So only proteins that carry a special ER signal sequence are imported into the ER. The signal sequence is recognized by the signal recognition particle, or the SRP, which the SRP, like in the thylakoid membrane transport, is almost the same, which binds with the growing polypeptide chain and the ribosome and directs them to a receptor protein on the cytosolic surface of the rough endoplasmic membrane. Okay, so this binding to the ER membrane initiates a translocation process that threads a loop, of polypeptide chain across the ER membrane through the hydrophilic core of a protein translocator. So soluble proteins that are distinct for the ER lumen for secretion or transferred to the lumen of other organelles pass completely through the ER lumen, especially if they are bound to other destinations via vesicular transport. So there are hydrophobic portion of the protein that can act either as start transfer or stop transfer signals during the translocation. So when a polypeptide contains multiple alternating start transfer and stop transfer signals, it will pass back and forth, across the bilayer multiple times as a multipass transmembrane protein. So the asymmetry of protein insertion and glycosylation to ER establishes the sidedness of the membrane of all the other organelles that the ER supplies of membrane proteins. Okay, so that is how a transmembrane protein, a protein that is embedded within the membrane of the ER, is actually synthesized. Okay, so that ends our lesson for today. So I hope you will review all the topics that we have discussed. In order to help you review these lessons or these different topics, you may want to answer these different questions. What are the different types of protein transport mechanisms? So we know that there are three. And how do they differ from each other? Okay, what is a sorting signal? How about a sorting receptor? How are proteins transported from the cytosol to the different cellular components? So how do they are how proteins transported from the cytosol to the nuclear membrane, to the cytoplasm, to the ER, to the mitochondria, to the chloroplast, to the peroxisomes? So you should be able to determine the differences. And how is gated transport different from transmembrane transport? So you should understand what are the examples of the transport mechanisms that are categorized as gated transport and transmembrane transport. Okay, so that ends our lesson for today. So if you have any questions, you can contact me or your lecturer through their provided email address. You can also directly key in your questions through the official group chat of our class. So thank you very much and see you again in our next lesson.